everyone, welcome to Calcium Video. Today we're going to discuss a very interesting topic which is lipoprotein metabolism. At first we need to know what is lipoprotein. From the name we can guess that lipoprotein is composed of lipid and protein, so it's a complex lipid. The protein portion of lipoprotein has a special name which is called apolipoprotein or simply apoprotein. By definition, lipoproteins are spherical macromolecular complex which is composed of lipid and specific protein called apolipoprotein. Now let's see how lipoproteins actually look like. This is the actual face of lipoprotein. In this figure, we can see that there is an inner core and an outer superficial layer. The inner core is composed of triacylglycerol and cholesterol ester, and the superficial layer is composed of phospholipid, free cholesterol, and apoproteins. This structure of lipoprotein is a very important breathing question, and you can easily answer this figure. From this figure, we can see that the inner core is composed of hydrophobic lipids, that means the lipids which are afraid of water. On the other hand, the phospholipid and free cholesterol are amphipathic lipid. That means they have both hydrophilic end and hydrophobic end. They are oriented in such a way that the hydrophilic end faces outwards and hydrophobic end faces inwards. That makes it soluble in water. We know that lipids are insoluble in water, so they can be transported in the blood alone. Lipoproteins are here to transport lipids from one place to another among various tissues and organs. So from this structure, we can know that the lipid portion of lipoprotein are triacylglycerol, phospholipid, cholesterol ester, and free cholesterol. And the protein portion is apoprotein. There are 5 types of apoproteins that are ApoA, ApoB48, ApoB100, ApoC, and ApoE. Different lipoproteins has different type of apoproteins in them. The ApoB48 and ApoB100, both of them has ApoB gene. But the difference is that the ApoB100, which is produced in the liver, has their B gene 100% coded. On the other hand, the ApoB48, which is produced in the intestine, has the B gene 48% coded. So that's the reason they differ from each other. The function of apoprotein is a very important written question. Let's see their function. They act as a structural component of lipoprotein and they maintain the structural stability of lipoprotein. They give a hydrophilic character to the lipoprotein particles to help their transport in the aqueous plasma. They also act as cofactor for enzymes. That means that they activate the enzymes. For example, ApoC2 activates lipoprotein lipase enzyme. ApoA2 activates hepatic lipase enzyme. Some of the apolipoproteins act as inhibitor of enzymes such as ApoC3 and ApoA2 acts as inhibitor of lipoprotein lipase. So you can see ApoC2 is the activator of lipoprotein lipase and ApoC3 and ApoA2 is the inhibitor of lipoprotein lipase. They also act as ligand to recognize lipoprotein receptor on cell surface for the uptake of lipoprotein, such as ApoB100 and ApoE acts as ligand for LDL receptor, and ApoA acts as a ligand for HDL receptor. So now, as you are clear about the structure of lipoprotein, let's see the type of lipoprotein. According to density, there are 4 types of lipoprotein. From low density to high density, there are chylomicron, very low density lipoprotein, low density lipoprotein, and high density lipoprotein. The chylomicron is originated in the intestine and its function is to transport triacylglycerol from the intestine to the peripheral tissue. The very low density lipoprotein is originated in the liver and their function is to transport triacylglycerol from the liver to the peripheral tissue. The low density lipoprotein is originated in the blood from VLDL and their function is to transport cholesterol into the peripheral tissue. And finally, high density lipoprotein. They are originated in the liver and their function is to transport cholesterol from the peripheral tissue to the liver. Now let's discuss the metabolism of individual lipoprotein. First comes chylomicron. If you have watched our video about lipid digestion and absorption, you might remember that the end product of lipid digestion in the intestine interact with an apoprotein called ApoB48 and assemble themselves into chylomicron, which is secreted in the circulation via the lymphatic system. So this is the nascent chylomicron in the circulation. This is called nascent because it's not matured. It needs more apoproteins to be fully matured. In the circulation, there is also HDL, which is the kindest lipoprotein. It donates its apoprotein to other lipoproteins when they need it. So here, HDL donates its ApoC and E to the nascent chylomicron. Now, the nascent chylomicron has triacylglycerol, cholesterol ester, free cholesterol, ApoB48, ApoC, and ApoE, and it's totally matured now. As chylomicron passes in the circulation, the ApoC2 in the chylomicron activates the lipoprotein lipase enzyme, which is situated at the capillary wall of the peripheral tissues, especially adipose tissue, skeletal muscle, and cardiac muscle. This lipoprotein lipase enters into the chylomicron and it degrades the triacylglycerol into fatty acid and cholesterol. The fatty acid enters into the cell where it is stored in the adipose tissue and used up as energy in skeletal muscle and cardiac muscle. On the other hand, the glycerol goes into the liver. As the major portion of chylomicron was triacylglycerol and it is degraded, the chylomicron reduces in size. Moreover, the ApoC is returned to the HDL. The remnant portion is called chylomicron remnant, which is cholesterol rich. This chylomicron remnant now goes into the liver and the hepatocytes degrades the chylomicron remnant by the process of endocytosis, and the ApoE inside of the chylomicron remnant helps the hepatocyte to recognize the cell. And this is the metabolic fate of the chylomicron. Let's recapitulate the process. The nascent chylomicron is secreted in the circulation from the intestine. Then the nascent chylomicron becomes matured by getting ApoC and ApoE from HDL. The ApoC2 in the chylomicron activates lipoprotein lipase and lipoprotein lipase degrades triacylglycerol into fatty acid and glycerol, and fatty acid goes into the cell and glycerol goes into the liver, and then the remnant portion is called chylomicron remnant, which goes into the liver where it is finally degraded. So this is how the chylomicron transports triacylglycerol from intestine to the peripheral tissue. The desired level of chylomicron is 100 to 200 mg/%. Now let's come to the VLDL metabolism. The function of VLDL is quite similar to chylomicron, except it transports the lipids which are produced in the liver. We know that the lipids which are synthesized inside of our body are called endogenous lipid. So the function of VLDL is to transport endogenous lipid from the liver to the peripheral tissue. Let's see how it's metabolized. The lipids which are synthesized in the liver interact with an apoprotein called ApoB100 in the liver and forms nascent VLDL. Nascent VLDL is secreted in the circulation from the liver through the process of exocytosis. This is the nascent VLDL in the circulation. It needs more apoproteins to be matured. Same as previous, the HDL donates its ApoC and ApoE to the nascent VLDL, and then the nascent VLDL is finally matured. While passing through the circulation, the ApoC2 in VLDL will activate the lipoprotein lipase enzyme from the peripheral tissue. The lipoprotein lipase enzyme will degrade the triacylglycerol of VLDL into fatty acid and glycerol. As the major portion of VLDL is now degraded, it will reduce in size. Moreover, it will return the ApoC and ApoE to the HDL. The remnant portion is cholesterol rich and which will ultimately convert into intermediate density lipid or IDL. This IDL acts as a precursor of LDL. That means LDL will be produced from IDL. So this is how VLDL is metabolized. Let's recapitulate the process. The nascent VLDL is secreted from the liver into the circulation. The VLDL gets matured by getting ApoC and ApoE from HDL. VLDL passes through the circulation and lipoprotein lipase degrades the triacylglycerol of VLDL, and the remnant portion will ultimately convert into intermediate density lipid, which will turn into LDL. So this is how VLDL transports endogenous lipids from the liver to the peripheral tissue, and the desired level of VLDL is less than 40 mg%. Easy, right? Now let's discuss LDL metabolism. LDL is produced from VLDL in the circulation, and their function is to transport cholesterol in the peripheral tissue. Let's see how it is metabolized. The first step is the transformation of IDL into LDL. We know that IDL is rich in cholesterol, but it still has some triacylglycerol. In the circulation, IDL receives cholesterol ester from HDL in exchange of triacylglycerol. The remaining triacylglycerol in IDL will be degraded by hepatic lipase into fatty acid and glycerol. With this change, IDL gradually transforms into LDL, which is rich in cholesterol ester and also contains ApoB100. In the circulation, the ApoB100 in the LDL will activate the LDL receptors, which are situated in the liver and peripheral tissue. After LDL receptors are activated, they will uptake LDL inside of the cell by the process of endocytosis. Inside of the cell, LDL will release its cholesterol in the cell, which can either be stored in the cell as the form of cholesterol ester, or the cell may use it for membrane synthesis, steroid synthesis, or bile acid synthesis. The remaining portion of LDL is uptaken by macrophage, which will be finally degraded, and that is the metabolic fate of LDL. Let's summarize the process. At first, IDL transforms into LDL by transferring the triacylglycerol to HDL in exchange of cholesterol ester. This cholesterol ester rich LDL will be uptaken by the liver and peripheral cells, which is mediated by LDL receptor, and inside the cell, LDL will offload its cholesterol, which will be used in the cell for various purposes, and the remaining portion will be uptaken by macrophages. LDL is called bad cholesterol because it increases the cholesterol level in the peripheral tissue, and we know that the increased amount of cholesterol level increases the risk of atherosclerosis. If there is excessive accumulation of LDL, Macrophage loaded with cholesterol turns into foam cell, which initiate atherosclerotic plaque formation. This increases the risk of coronary artery disease, cerebrovascular disease, etc. So there is adverse effect of LDL in our health. So that's why the level of LDL should be kept in a minimum level less than 130 mg/cent. Now let's discuss HDL metabolism. HDL transports cholesterol from the peripheral tissue back to the liver. Let's see how they do this. HDL is produced by the hepatic cells and intestinal cells as the form of nascent HDL. Nascent HDL contains apoprotein ApoA, ApoC, ApoE, and the lipid portion is mainly cholesterol ester. After formation, nascent HDL is secreted in the blood. While traversing through the circulatory system, HDL get attached with the HDL receptor situated on the peripheral cell and it uptakes cholesterol from the cell. While coming to HDL, the cholesterol converts into cholesterol ester by the enzyme lecithin cholesterol acyltransferase. Gradually, after accumulating cholesterol ester, nascent HDL gets matured into a spherical form. This is called HDL3. Subsequently, HDL3 transfers part of its cholesterol ester to other ApoB containing circulating lipoproteins such as VLDL, IDL, and LDL in exchange of triacylglycerol. After accumulating triacylglycerol, the HDL3 gets even bigger and now it's called HDL2. This HDL2 will go back to the liver where it offloads its cholesterol ester. In the liver, the cholesterol may be excreted with bile or it may be converted to bile acid or maybe cholesterol will be repackaged into VLDL. There's an interesting thing. After offloading cholesterol ester, the hepatic lipase degrades the remaining triacylglycerol of HDL2 and then the liver will convert the HDL2 into HDL3 and it is secreted in the circulation and this process will begin to continue. This interchange between HDL3 and HDL2 is called HDL cycle. The transport of cholesterol from the peripheral tissue back to the liver through HDL cycle is called reverse cholesterol transport. It can be done in two routes. In the direct route, the nascent HDL uptakes cholesterol from the peripheral tissue and it directly delivers it into the liver. And in the indirect route, nascent HDL uptakes cholesterol from the peripheral cells. Then the HDL3 will transport the cholesterol ester to other lipoproteins VLDL, IDL, and LDL, and then those lipoproteins will go back to the liver. As the HDL clears off cholesterol from the peripheral cells, HDL is called cholesterol scavengers. This is how, by the process of HDL cycle, HDL uptakes cholesterol and returns them to the liver. Let's recapitulate the process. At first, nascent HDL is formed by the hepatic cells and intestinal cells and it's secreted in the circulation. HDL get attached with the receptor from peripheral cells and they take up cholesterol from the cells. After accumulating cholesterol ester, they get in a spherical form called HDL3 and subsequently they transfer some cholesterol ester to other lipoproteins in exchange of triacylglycerol. And after accumulating triacylglycerol, they get even larger, which is called HDL2, which ultimately goes back to the liver where it offloads its cholesterol. And this is how the reverse cholesterol transfer process occurs. This reverse cholesterol transport process is very important for our body. We know that Excessive accumulation of cholesterol increases the risk of atherosclerotic disorders. HDL takes up the cholesterol from peripheral tissue back to the liver, and this is how they reduce the serum cholesterol level in our body, and ultimately they reduce the risk of atherosclerotic disorders. This is why HDL is called good cholesterol. It has also antioxidant activity. So that's why the level of HDL should be maintained greater than 40 mg/cent in our body. So that was the metabolism of all four lipoproteins. I hope that you clearly understood the process. Still, if you have any question, don't hesitate to comment in our comment box. If you think this video helped you, don't forget to like this video and subscribe our channel for more amazing content. Thanks a lot for watching.